From the name, it is quite evident that polysaccharides are made up of many monosaccharide units which are joined by bonds called glycosidic bonds. They are covalent bonds. Polysaccharides can be classified as homopolysaccharides and heteropolysaccharides. The important homopolysaccharides are starch, glycogen, chitin and cellulose, and important heteropolysaccharides are pectin, murin, agar, agar-agar, agarose, alginate, etc. The list of homopolysaccharides and heteropolysaccharides is quite long, and these polysaccharides perform very important functions for the maintenance of the cell survival depends on them. So, what are homopolysaccharides? Homopolysaccharides means the polysaccharide is made up of same type or only one type of monosaccharides, and hetero means different, at least two different types of monosaccharides are there in the heteropolysaccharides. So, the homopolysaccharides or heteropolysaccharides can be classified again as the energy reserves or structural part. So, homopolysaccharides are starch, glycogen. These two are the storage forms of glucose in the cell. Starch is the storage form of glucose in the plant cells, and glycogen is the storage form of glucose in the animal cells. And cellulose and chitin form the structural polysaccharides. They maintain the structure. Chitin is is found in the exoskeletons of spiders, then crustaceans and other insects, and also in some yeast or fungal cell wall, and cellulose is a main component of the plant cell wall. So, how are starch and glycogen linked in a polysaccharides? Starch and glycogen more or less are same, but there is a difference as the position of the glycosidic bonds. The glucose units of starch are joined by alpha 1,4 glycosidic bonds, and also there are branch points in the entire molecule, and the branch points are at alpha 1,6 glycosidic bonds, and these comprises of the alpha 1,4 and alpha 1,6 glycosidic bonds, and the entire molecule of starch is formed. So, there are two types of starch, amylose and amylopectin, and amylose is the unbranched type of starch and amylopectin is the branched form. So, in amylose, the unbranched type, that means only 1,4 glycosidic bonds are there, and in amylopectin both types of glycosidic bonds are present. And glycogen is also a polymer of glucose, and only alpha 1,4 glycosidic bonds are there. These are the structures of amylopectin, as shown. And other important structural polysaccharides are cellulose and chitin. So, cellulose is also made up of glucose, but instead of alpha 1,4 glycosidic bonds, it is made up of beta 1,4 glycosidic bonds. And also chitin. Chitin is also formed by the beta 1,4 glycosidic bonds, and N-acetyl glucosamine and N-acetyl glucosamine, that means homopolysaccharide, they are joined by the beta 1,4 glycosidic bonds. These are shown that chitin is the structural component of the exoskeletons, and also it is found in cell wall of algae and fungi. Other homopolysaccharides can also be mentioned, such as xylan. It is a polysaccharide. It is made up of xylose monosaccharides. The general name of the homopolysaccharides is glycan, because they are formed by the joining of glycosidic bonds. So, general name is glycans, and xylan is therefore xylose containing polysaccharide, and the bond is beta 1,3. Other examples are callose. Callose is also found in new green plants, and it is a glucan. Therefore, the monosaccharides which comprise the callose are glucose, and the beta 1,3 linkage is there. Caesophyllan is another yeast cell wall component. and it is also a glucan having beta 1,3 and beta 1,6 glycosidic bonds. At some positions there are beta 1,6. Paramylon, it is found in euglena. Paramylon is also a glucan, beta 1,3 glycosidic bond is there, and the paramylon is a storage polysaccharide in euglena. Apart from them, there are mannans. Mannans means polysaccharides having mannose subunits or mannose monosaccharides. Alpha mannans and beta mannans. That means alpha glycosidic bond in alpha mannan, beta glycosidic bond in beta mannans, and in case of alpha mannans, the presence of alpha mannans is in the yeast cell wall. Beta mannans are present in microfibrils of algal cell wall. Then dextrans. Dextrans are a family of polysaccharides which are linked by alpha 1,6 glycosidic bonds, and the component subunit is glucose, and also there are alpha 1,3 branch points, and dextrans are produced by bacteria. Some of these dextrans are chemically cross-linked or chemically modified to form beads, and these are used in purification or separation of biomolecules like proteins. Then inulin. Inulin is a storage polysaccharide found in sweet potatoes, and the units or monosaccharide units is fructose. The structural difference has been shown over here between the starch and glycogen and cellulose. Cellulose are shown as chains. All the beta 1,4 glycosidic bonds are so oriented that a chain of cellulose can be prepared, and these chains, the chains of cellulose are linked to each other by hydrogen bonds, and these long chains run parallel, and they are responsible for the high tensile strength of the plants. But in starch and glycogen, the structure is not arranged in a chain form, and this is the proposed structural model of cellulose, and the cellulose fibers are extended as I have already said, and there are intra-chain hydrogen bondings also, and between two chains also hydrogen bonding exists. Oh, about heteropolysaccharides. Heteropolysaccharides, these are pectin, murin, agar-agar, alginate, and these heteropolysaccharides are classified on the basis of their presence in either plant cells or animal cells or bacterial cells. These are the important heteropolysaccharides of the animal body: hyaluronan, chondroitin sulfate, keratan sulfate, dermatan sulfate, and heparan sulfate. As the name suggests, all these four are sulfated, and these sulfated means they are charged. And hyaluronan is a straight chain compound heteropolysaccharide, and here these are all associated with proteins forming proteoglycans. So these proteoglycans play a very important role in maintaining the integrity of the connective tissues in our body. And hyaluronan, or it is also said hyaluronic acid, it is a derivative, hyaluronan salt, hyaluronic acid, and these form the ground substance of the connective tissues. And chondroitin sulfate again and dermatan sulfate, these are important components of proteoglycans, and as the name suggests that dermatan sulfate is found in skin, chondroitin sulfate is found in cartilage, keratan sulfate is also found in cartilage, cornea and bones, and heparan sulfate is most popular or most ubiquitous of all these heteropolysaccharides. They are found on the cell lining, epithelial cellular lining and also in the brain. This heparan sulfate can be found, and all these are important in respect to modifications, in respect to the positions of their modifications, that is the sulfonation sites. They are all different, and the hyaluronan is formed by the beta 1,4 linkage with glucuronic acid and N-acetyl glucosamine. Glucuronic acid, that means there is an acidic component here, and beta 1,4 glycosidic bonds are formed, and chondroitin sulfate and dermatan sulfates, these are apimers. Apimers, that means the conformation of any one carbon is different. That means OH conformation or anything conformation is different. So at only one carbon, and chondroitin sulfate and dermatan sulfate have differences in the carbon 5, and chondroitin sulfate is present in cartilage. The main components are beta D glucuronate and N-acetyl galactosamine. N-acetyl galactosamine, chondroitin sulfate, and this N-acetyl galactosamine can be sulfated, and the position of the sulfate makes the naming something different. That means chondroitin 4 sulfate or chondroitin 6 sulfate, because 4 carbon the group has been sulfated, and in 6 sulfate, that means 6 carbon group has been sulfated. And dermatan sulfate is also sulfated after the arrangement of this chain, and it is also the position of the sulfate group is different. Instead of beta D glucuronate in case of chondroitin sulfate, dermatan sulfate has L iduronate instead of glucuronate, and beta, instead of beta form, it is alpha form, but the same N-acetyl galactosamine is same in both these cases. In case of keratan sulfate, the components are beta 1,4 linked galactose and N-acetyl glucosamine, and in case of heparan sulfate, the components are D-glucuronate and N-acetyl glucosamine. The bond is beta 1,4, but there are heparan sulfate forms a family of molecules where it may be different in the beta conformation, beta 1,4, beta 1,3, they may be different. There are several similar molecules and performing the same function. These are the heteropolysaccharides of the plants. Carrageenans, they form the gel material, gel forming material. Pectin is also a jelly like material, quite prevalently used in food industry. Alginate is found in the cell wall of marine algae. Then agarose is a gelling matrix. It is very popularly used in the biological laboratories for several experimental purposes. Pectin is a polysaccharide where polyglucuronates, glucuronic acid that is charged and polymeric forms. In sometimes this polymer contains L-rhamnose. Rhamnose is a deoxy sugar. But though the major component is glucuronates in pectin, and they perform several functions in the plants also. Apart from them, these are also used for our several industrial purposes or laboratory purposes. These are the structures of some of the polysaccharides. Agarobiose is one of the components of agar. Apart from heteropolysaccharides of plants and animals, bacterial heteropolysaccharides also play a very important role in maintaining the structure of the cell, bacterial cell. And the major compound involved in maintaining the structure is peptidoglycan or murin. The list also shows the names of the prominent polysaccharides, homo and heteropolysaccharides, of the prominent polysaccharides that are found in nature, but the list is too short. We must discuss about the bacterial cell wall, because the bacterial cell wall is made up of the polysaccharide units arranged in a chain. So, the constituents are N-acetyl D-glucosamine and N-acetyl D-muramic acid, and these alternating chains of N-acetyl D-glucosamine and N-acetyl D-muramic acid are arranged as shown in the figure. These are the compounds, polysaccharides chains, and each parallelly several chains are running, and these chains are related to each other or linked to each other by a peptide chain and a pentaglycine bridge. So, peptide means they are formed from amino acids, and glycine is again another amino acid, but this is a pentaglycine bridge. That means 5 glycine molecules are joined side by side, and these peptide chain and the pentaglycine bridge forms the connection between the chains of this alternating N-acetyl D-glucosamine and N-acetyl D-muramic acid, and this structure forms a meshwork like structure, network also we can call, around the bacterial cell. This is the main component of the bacterial cell wall, and without this bacterial cell wall the bacteria will not exist, cannot exist rather, and they are quite tough and porous network, and the porosity is maintained by this cross-linking of these chains, and porosity is also very necessary because for the transit of raw materials, nutrients inside the cell and waste products outside the cell. So, it has to be maintained in a porous network form. Another importance of this peptidoglycan or murin is that the extent of peptidoglycan in the entire bacterial population divides the bacterial population into 2 groups, gram positive and gram negative. So, for the classification of bacteria, we go to search how much or what is the extent of the magnitude of the peptidoglycan. In gram positive bacteria, the peptidoglycan layer is thick, and in gram negative bacteria it is quite thin compared to the gram positive. So, the gram positive bacteria have a thick peptidoglycan layer, but gram negative bacteria have a lipopolysaccharide layer apart from the thin peptidoglycan, and this lipopolysaccharide layer is present on the outer surface of the bacteria. So, what is the functional role of these lipopolysaccharides? This layer is in short called LPS. This LPS layer has a lipid molecules, and the polysaccharide has been associated with this lipid molecule. The polysaccharide, there are 3 zones: inner core, outer core and the O antigen. These polysaccharides, which they are actually projecting outwards from the cell, which are linked to the lipid, that is the lipid A, and this entire structure forms the lipopolysaccharide layer. So, very peculiar monosaccharide units, or unique, I must say, unique monosaccharides are present in the O antigens, and this O antigen, it has been shown that these variations in the sequence of the polysaccharides in the O antigen leads to several strains of bacteria, same bacteria but several strains, and this is called antigen because antibodies can be raised against this sequence or against this region of the bacteria. So, the important monosaccharides are paratose, then KDO. KDO is 2-keto-3-deoxyoctonate. This is a peculiar molecule monosaccharide, but they are joined by several types of linkages, alpha beta linkages, forming the O antigen chain. And the projecting ends of the lipopolysaccharide molecules consists of long carbohydrate chains with repeating units that have antigenic properties, and they are called O antigens. Now, several bacterial heteropolysaccharides are synthesized by the bacteria itself. Apart from these previous 2 cases, they are the structural components of the bacterial cell, and these are the byproducts of the bacteria secretion or bacterial metabolism. Some bacteria form gums, xanthan gum, these are sticky material, and acetan, these are also gums. So, xanthan gum is secreted and they have very important commercial uses. Xanthan gum is secreted by Xanthomonas campestris, acetan by Acidobacter xyleum. These are all polymeric compounds. They are the byproducts of the bacterial metabolism. Apart from bacterial, animal and plant heteropolysaccharides, a section has been kept aside that is called glycoproteins. That means here the carbohydrates play another different role, mainly biological recognition or maintenance of cellular integrity in specific regions, and also maintaining cell-cell communication or cellular adhesion. These molecules are very important, glycoproteins and proteoglycans. Now let us see what is a glycoprotein. Glycoproteins are the carbohydrate attached proteins. That means a carbohydrate is covalently attached to a protein to form a glycoprotein, and the bonds which are involved is N-linked glycosylation or O-linked glycosylation. In N-linked glycosidic bonds between proteins, the asparagine amino acid is involved, and in O-linked glycosidic bond formation, the serine residue of the protein is used, forming the O-linked N-acetyl or glycosidic bond, and in this case N-linked N-linked glycosidic bonds. And this shows the oligosaccharide has been completely attached to the asparagine, forming an entire moiety of oligosaccharide has been attached to asparagine. Here also the entire structure or entire oligosaccharide has been joined to asparagine, forming the glycoproteins. There are three classes of glycoproteins. In the first class, the protein constituent is the largest component by weight. Here the protein component is predominant, and these versatile biomolecules play a variety of biochemical roles. What are they? They are the components of the cell membrane, where they take part in processes such as cell adhesion, binding of sperms to eggs. Some of them are hormones. The main such hormone is erythropoietin. Erythropoietin is very important for the formation of blood cells. Many of them are involved in cellular growth and differentiation, and many glycoproteins of this class, that is the first class, are involved in host pathogen. The pathogen may be bacteria or virus, host pathogen interaction. The second class of glycoproteins, they are the proteoglycans. The protein component is conjugated to glycosaminoglycans. So here the carbohydrate part is prominent, and the carbohydrates are the major components of these glycoproteins. The difference is quite evident. About 95% by weight of these glycoproteins is carbohydrates. They function as lubricants and structural components in connective tissues, mediating the adhesion of cells to the extracellular matrix and binding to factors that stimulate cell proliferation. These are the examples, a few examples already mentioned, the glycosaminoglycans, which when associated with proteins gives the proteoglycans. The glycosaminoglycans plus proteins giving the proteoglycans. The functions are the extracellular spaces in the connective tissues such as cartilage, tendon, skin and blood vessel walls consists of collagen fibers, and they are mainly embedded in a gel-like matrix known as the ground substance. It is present everywhere in our body, and this ground substance is composed of the glycosaminoglycans. These are slimy, mucus-like, which is the reason for their high viscosity and elasticity. And the proteoglycans are the glycosaminoglycans which are covalently or non-covalently bound to proteins in the basement membranes and in cell surface membranes, and these aggregated and covalently or non-covalently to form the diverse group of proteoglycans. Proteoglycans have a core protein to which at least one glycosaminoglycan chain is covalently linked. A chain of the glycosaminoglycans has been linked with the core protein and the proteoglycan, these are the proteoglycan subunits. They are joined with a link protein, with the help of a link protein to the molecule, one molecule or several molecules of hyaluronan. This entire structure has been depicted in the next picture. This is the entire structure of the proteoglycan, covering the core protein, subunit structure. These are the subunit structure, and these subunits are formed by the joining of these glycosaminoglycans. These are all called glycosaminoglycans because these are aminated. And this is the link protein. Link protein is also connected with the hyaluronic acid chain. And this is the electron micrograph picture. Third class or the class 3 glycoproteins, they form a distinct family of O-linked glycoproteins and are called mucins. The mucins are predominantly carbohydrates similar to the class 2, and the N-acetyl-galactosamine is present prominently. They serve as lubricants. Mucins are present in saliva and in mucous secretions. Mucins often serve protective functions. Some other mucins contain large oligosaccharides which often carry blood group determinants. They are the examples, concrete examples of mucin-like structures, and these difference in the carbohydrate groups give rise to the difference in the blood groups of different individuals. So, to summarize, these are the major features or major functions that the carbohydrates play. And the one thing which is very interesting is that all these carbohydrates are synthesized in the cell, and you can think that the monosaccharide is such a small unit and they comprise such a complex array of carbohydrates.
Ma'am, I would like to know what is the difference between heparan and heparin.
Heparan, the structure has been shown here, and in heparan there is a beta bonds and the number of sulfate groups are much less. But in case of heparin, the bond is alpha linkage and the number of sulfates are many, sulfation, and the property is also different because heparin has an anticoagulant property, but heparan is present in the cell membranes.